Hello everybody, so from this lesson on, we will be learning about the drugs that act on the autonomic nervous system. The autonomic nervous system, otherwise known as the ANS, is the major involuntary, unconscious, and automatic portion of the nervous system, and contrasts in several different ways with the somatic or voluntary nervous system. The anatomy, neurotransmitter chemistry, receptor characteristics, and functional integration of the autonomic nervous system are discussed in this chapter. Major autonomic drug groups are discussed in chapters 6, 7, 8, and 9. The motor or efferent portion of the autonomic nervous system is a major pathway for information transmission from the central nervous system to the involuntary affected tissues like smooth muscles, cardiac muscles, and exocrine glands. Its two major subdivisions are the parasympathetic autonomic nervous system, otherwise known as PANS, and the sympathetic autonomic nervous system, otherwise known as SANS. The term autonomic is derived from the root auto, meaning self, and nomos, meaning law. The autonomic nervous system regulates the functions of the involuntary organs, which include the heart, the blood vessels, the exocrine glands, and the visceral organs. The autonomic nervous system consists of a two-neuron efferent chain. Postganglionic axons terminate in varicosities, swelling containing synaptic vesicles, which release the transmitter into the extracellular space surrounding the effector cells. So, how do we define different neuron types in the autonomic nervous system? Well, the autonomic nervous system is usually subdivided into three different divisions: the sympathetic, the parasympathetic, and the enteric. The sympathetic system was designed as adrenergic for the adrenaline-like actions resulting from sympathetic nerve activation, and the parasympathetic system was designed as cholinergic for the acetylcholine-like actions. The sympathetic division is said to preside over the use of metabolic resources and emergency responses of the body, just like an accelerator, and is more commonly known as the fight or flight response. The parasympathetic division presides over the restoration of the body's reserves and elimination of waste products, just like a brake, and is known also as rest and digest. In reality, most of the organs supplied by the autonomic nervous system receive both parasympathetic and sympathetic innervations. Each division of the autonomic nervous system has specific effects on organ systems. These effects are summarized in the table currently on the slide 63 and should be memorized. Dually innervated organs, such as the iris of the eye and the sinoatrial node of the heart, receive both sympathetic and parasympathetic innervation. The pupil has a natural intrinsic diameter to which it returns to when the influence of both divisions of the autonomic nervous system are removed. Apharmacologic ganglionic blockade, therefore, will cause it to move back to its intrinsic and original size. Similarly, the cardiac sinus node pacemaker rate has an intrinsic value, about 100 to 110 per minute, in the absence of both autonomic nervous system inputs. So how will these variables change, increase or decrease if the ganglia are blocked? The answer is predictable if you know which system is dominant. For example, both the pupil and, at rest, the sinoatrial node are dominated by the parasympathetic system. The resting pupil diameter and sinus rates are therefore under considerable PANS influence. Thus, a blockade of both systems with removal of the dominant PANS and non-dominant SANS effects result in mydriasis and tachycardia. The synthesis, storage, release, receptor interactions, and termination of action of the neurotransmitters all contribute to the action of autonomic drugs. Acetylcholine, ACH, is the primary transmitter in all autonomic ganglia and at the synapses between parasympathetic and postganglionic neurons and their effector cells. It is also the primary transmitter at the somatic or voluntary skeletal muscle neuromuscular junction. Acetylcholine is synthesized in the nerve terminal from acetyl, CoA, produced in the mitochondria, and choline, transported across the cell membrane by the enzyme choline acetyltransferase, or CHAT. The rate-limiting step is probably the transport of choline into the nerve terminal. This transport can be inhibited by the research drug hemicholine. Acetylcholine is actively transported into its vesicles for storage by the vesicle-associated transporter, VAT, or VAT. This process can be inhibited by another research drug, Vesamicol. Release of transmitter stores from vesicles into the nerve ending requires the entry of calcium through calcium channels and the triggering of an interaction between snare proteins. This interaction results in the docking of the vesicle to the terminal membrane, and, with an influx of calcium, fusion of the membranes of the vesicles with the nerve-ending membranes, the opening of a pore to the extracellular space, and the release of the stored transmitter. The several types of botulinum toxins enzymatically alter synaptobravin, or one of the other docking or fusing proteins, to prevent the release process. The action of acetylcholine in the synapse is normally terminated by metabolism to acetate and choline by the enzyme acetylcholinesterase in the synaptic cleft. The products are not excreted but are recycled in the body. Inhibition of acetylcholinesterase is an important therapeutic effect of several drugs. Muscarinic receptors. Muscarine is a natural product found in certain mushrooms. Muscarine was first isolated in 1869, and it was the first parasympathomimetic substance ever studied, and causes profound activation of the parasympathetic nervous system. As their name suggests, these receptors respond to muscarine, an alkaloid, as well as acetylcholine. The effects of the activation of these receptors resembles those of postganglionic parasympathetic nerve stimulation. Muscarinic receptors are located primarily on autonomic effector cells, including heart, vascular endothelium, smooth muscle, presynaptic nerve terminals, and exocrine glands. Evidence including their genes has been found for 5 subtypes, of which 3 appear to be important in peripheral autonomic transmission. All 5 are G protein coupled receptors. Nicotinic receptors are located on ion channels and respond to acetylcholine and nicotine, another acetylcholine mimic, but not to muscarine, by opening the channel. The 2 major nicotinic subtypes are located in ganglia and in skeletal muscle end plates. The nicotinic receptors are the primary receptors for transmission at these sites. This table contains the characteristics of the most important cholineceptors in the peripheral nervous system. Because of the number of steps in the transmission of autonomic commands from the central nervous system to the effector cells, there are many sites at which autonomic drugs may act. These sites include the central nervous system centers, the ganglia, the postganglionic nerve terminals, the effector cell receptors, and the mechanisms responsible for transmitter synthesis, storage, release, and termination of action. The most selective effect is achieved by drugs acting at receptors that mediate very selective actions, tables 6 to 4. Many natural and synthetic toxins have significant effects on autonomic and somatic nerve function. Drugs that block the synthesis of acetylcholine, e.g. hemicolinium, its storage, e.g. versamicol, or release, e.g. botulinum toxin, are not very useful for systemic therapy because their effects are not sufficiently selective, i.e. pans and sans ganglia and somatic neuromuscular junctions all may be blocked. However, because botulinum toxin is a very large molecule and diffuses very slowly, it can be used by injection for relatively selective local effects. Adrenergic transmission. Noradrenaline, Na, norepinephrine, Ne, is the primary transmitter at the sympathetic postganglionic neuron effector cell synapses in most tissues. Important exceptions include sympathetic fibers to thermoregulatory sweat glands and probably vasodilator sympathetic fibers in skeletal muscle, which releases acetylcholine. Dopamine may be a vasodilator transmitter in renal blood vessels, but norepinephrine is a vasoconstrictor of these vessels. Synthesis and storage. The synthesis of dopamine and norepinephrine requires several steps. After transport across the cell membrane, tyrosine is hydroxylated by tyrosine hydroxylase, the rate-limiting step to DOPA, decarboxylated to dopamine, and inside the vesicle, hydroxylated to norepinephrine. Tyrosine hydroxylase can be inhibited by metirosine. Norepinephrine and dopamine are transported in vesicles and stored there. Monoamine oxidase, MAO, is present on mitochondria in the adrenergic cell nerve ending and inactivates a portion of the dopamine and norepinephrine in the cytoplasm. Therefore, MAO inhibitors may increase the stores of these transmitters and other amines in the nerve endings. The vesicular transporter can be inhibited by reserpine, resulting in the depletion of transmitter stores. Release and termination of the action. Dopamine and norepinephrine are released from their nerve endings by the same calcium-dependent mechanism responsible for acetylcholine release. Termination of action, however, is quite different. Metabolism is not responsible for termination of action of the catecholamine transmitters, norepinephrine and dopamine. Rather, diffusion and reuptake, especially reuptake 1 by the norepinephrine transmitter NET, or the dopamine transmitter DAT, reduce their concentration in the synaptic cleft and stop their action. Outside the cleft, these transmitters can be metabolized by MAO and catechol O methyltransferase, COMT, and the products of these enzymatic reactions are excreted. Determination of the 24-hour excretion of metanephrine, normetanephrine, 3-methoxy-4-hydroxymandelic acid, VMA, and other metabolites provides a measure of the total body production of catecholamines, a determination useful in diagnosing conditions such as pheochromocytoma. Inhibition of MAO increases stores of catecholamines and has been both therapeutic and toxic potential. Drug effects on adrenergic transmission. Drugs that block norepinephrine synthesis, e.g. metirosine, or catecholamine storage, e.g. reserpine, or release, guanethidine, have been used in treatment of several diseases, e.g. pheochromocytoma, or hypertension, because they block sympathetic but not parasympathetic functions. Other drugs promote catecholamine release, e.g. the amphetamine-like agents, and predictably cause sympathomimetic effects. Adrenergic receptors are classified as either alpha or beta, subclassified as either alpha or alpha 2 and beta 1, beta 2, or beta 3 receptors. Alpha receptors. These are located on vascular smooth muscle, presynaptic nerve terminals, blood pallets, fat cells, and lipocytes, and neurons in the brain. Alpha receptors are further divided into two major groups, alpha 1 and alpha 2. These two subtypes constitute different families and use different G-coupling proteins. Beta receptors. These receptors are located on most types of smooth muscle, cardiac muscle, some presynaptic nerve terminals, and lipocytes, as well as in the brain. Beta receptors are divided into three major subtypes, beta 1, beta 2, and beta 3. These subtypes are similar in their use of the G-coupling protein. Dopamine receptors. Dopamine, DA receptors, are a subclass of adrenoceptors, but with rather different distribution and function. Dopamine receptors are especially important in the renal and splanchnic vessels and in the brain. Although at least 5 subtypes exist, the D1 subtype appears to be the most important dopamine receptor on peripheral effector cells. The D2 receptors are found in presynaptic nerve terminals. D1, D2, and other types of dopamine receptors also occur in the central nervous system. Summary of parasympathetic neurons and synapses. So we've got the preganglionic neurons, which are long. Synapses with postganglionic neurons at or near the organ releases acetylcholine to activate nicotinic receptors on postganglionic neurons. Postganglionic. Short synapse on the target organ releases acetylcholine to activate muscarinic receptors on the target organ. Summary of sympathetic neurons and synapses. So we've got the preganglionic neurons, short. Synapse with postganglionic neurons near spinal cord releases acetylcholine to activate nicotinic receptors on postganglionic neurons. We've got postganglionic neurons, long. Synapse on target organ releases norepinephrine to activate adrenergic receptors on target organs. Here is the checklist for what you should be able to complete once you have finished this chapter. And here are the references and sources for this presentation. Thank you.